For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 3, Episode 22 of Surfing the NASH Tsunami. Our preview of the 5th Global NASH Congress starts now.
This week on Surfing the NASH Tsunami.
I think for a long time, we've approached these tools quite simplistically, so it's nice to hear that we're being more strategic in understanding their outcomes.
There was a bit of information out at EASL last year and ARSL that the quality of diet actually improved the liver steatosis in preference to weight loss. All of these, as has been discussed already today, shed a new light on it's not always quantity of weight loss, it's actually the quality and how you lose it and what you lose it with, which may well benefit a lot of people.
The question is, which communities need to know what? I think it's a real challenge with emerging technologies for people to learn enough so that they're knowledgeable about them so that they can actually understand where they might be useful in their practice. That's quite a big ask for the sort of general, secondary, even tertiary care, clinical gastroenterologist, hepatologist.
I think comparing and contrasting in that way the nutritional aspect and weight loss, and there's a couple of sessions on weight loss with the drugs, it's almost a head-to-head comparison in the room.
Going back to Jörn's comment, how much of the science, the level you're talking about, do you feel physicians need to understand? A, physicians need to understand in order to be able to treat, and B, physician researchers need to understand in order to be able to do a better job on drug development and assessing what the results of their studies mean.
I'm excited to hear more about updates on gut microbiome and the association with disease. I think it's a completely underexplored aspect of the human genome. It's often been referred to as the second human genome, if not even the primary, because for every one cell of human cells that we have in the body, there's 10 cells of microbes.
I just went through the program to make sure I'm not missing anything. Key people from the companies that are reporting on their drug development program. There is Galmet present with Aram Cole. There is some data of semaglutide for combinations. There is the cirrhosis study that's been done by Galectin, and Paul Boots is going to talk about belapectin as an inhibitor. It's very interesting to hear from them what they think has been a breakthrough for their drug development program.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researcher and key opinion leader Professor Jörn Schattenberg, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and this week's guests, hepatology researcher and key opinion leader Professor Ian Rowe and Aged Diagnostic CEO Rachel Zayas, as they preview next month's 5th Global NASH Congress, this week on Surfing the NASH Tsunami.
Для всіх, хто цікавиться мережею NASH або загалом жировою хворобою печінки, ловіть хвилю.
Okay, so the first thing I want to do is I want to thank, again, our friend Anna Tokar for doing such a nice job of translating our opening line into I don't have to mangle pronunciation on a word-by-word basis. And as with previous weeks and future weeks, our thoughts and our hopes and our prayers with people of the Ukraine as they deal with this horrible situation in which they find themselves. Well, it's been a momentous week. Since we got together, Louise has flown home from Australia. Jörn was on vacation for a week, and he has returned. And we've located the mysterious Ian Rowe somewhere in the vicinity of Leeds. And we're even able to get Rachel Zayas to join us on a last-minute basis. So, hi, everybody. How are you all doing today?
All good.
Great.
Wonderful.
Quick icebreaker. Okay, one good thing, personal or professional, that's happened in the past week. Brave one, go first.
I just got back from my Easter break, as you mentioned, and first day in clinic today. There's so many things to catch up. I don't think I have all the backlog quite handled this evening. But many good things happened, of course, at the personal level. Being a week off, that's very relaxing and rewarding.
And it means you weren't in Mainz when Louise flew over.
Yeah, and of course, Mainz is not far from Frankfurt, so that happens every once in a while for everybody who's in the vicinity. And if you have layover, I'm happy to have a coffee or something.
I'll take you up on that one day. Obviously, I had a 3-week trip back to my home, my first home city of Sydney. But it was a great wedding with some friends, and it was great to catch up with everybody and have a sleepover, or only the Australians can do, where you tie all your motorboats together and you walk along the marlin boards between barbecues. So without falling in. So it was a very enjoyable and relaxing period away. And now I'm back to the world of fatty liver disease cascading up in the UK, as far as I can tell from my figures.
I'll jump in next. I'll go with a personal. I've played basketball the last 3 days in a row. It's been 70 something and sunny for the last 3 days in DC. And I play with a group of friends that I work with. And we've just been playing like children for the last 3 days, which is just incredible and healthy and social and lots of fresh air. So I've had a wonderful weekend.
Yeah, well, that sounds totally wonderful. I can't remember what 70 degrees looks like. We're into summer sports here, so it's cricket at between 6 and 8 degrees Celsius, which is fine, I think, if you're playing. But when you're standing watching for hours and hours on end, it is really, really cold. But it's still a highlight as my oldest son at 12 played his first senior cricket match this past weekend, took a wicket, and he's been delighted ever since.
Just takes one wicket to do it when you're 12, huh? That's great.
Yeah, well, I think when you take the wicket of an adult batsman who's on 39, he was a real success.
Oh, really?
Yeah.
That's fantastic. I remember from last year that your son is quite an accomplished young bowler, I guess. But that's amazing. And I'll commend Ian, because the last time he was on the podcast, he was worried that his beloved Newcastle Football Club was facing relegation. And now the question is whether they wind up in the top half of the EPL table or not. So if you see a smile cracking his face right now, 2/3 of it is his son and 1/3 of it is the Geordies. I'll go very quickly. I love this spring and summer because the town we live in is a small little town, charming and small. You can put your car away on Friday night and walk to everything you're going to do all weekend and not get in your car again until Monday or Tuesday. This is the first weekend of the year I got to do that. That's not quite true because we had my granddaughter on Saturday, so we had to drive her home to her parents. But except for that, we were not in the car all weekend, and we had a fantastic time. Friends, family, good weather. Saw a play yesterday, all good stuff. With that, let's dive into our content. What we're going to do today is we're going to review the 5th Global NASH Congress, which comes up at the end of May, 26th and 27th in London. We will be doing at least one day of live coverage from the conference, possibly two, probably one, and then probably a wrap-up episode at the end, but as media partners. But they've got so far 30 speakers on their list and a variety of different topics. And what I was hoping we would do today was, in one sense of the word, at least start at the beginning and go to the end, by which I mean start by they talk on their agenda page about 5 different categories of topics, and I'll just give you the headlines on those. In their summary, they discuss that their topics include recent updates in pathogenesis, preclinical and translational strategy, new targets for treatment of NASH fibrosis, and then non-invasive biomarkers and diagnostic tools, clinical development, clinical trial data, and then regulatory updates. So I want to kind of start at the top of the train and go to the bottom. So talk about the preclinical and the updates in pathogenesis first, and then we'll do the non-invasive clinical development second and regulatory third. And I would like one of you to pick a paper or a topic that you see in this agenda that you think might be particularly interesting, and just dive in. Mention what it is, what the headline is, why you think it might be interesting, and we'll go back to typical brave one go first.
Yeah, before we actually dive into the content, Roger, let me say, there's been a number of NASH meetings out there, and the 5th Global implicates, I think it had a break over the COVID year, but it's the 5th one on this. And I think I went to the first in person and then followed the other ones. For me, it was always very interesting because it had strong industry involvement. I mean, there are key speakers from the companies that run the clinical trials, and I think this is one of the specialities of that Congress. If I think about a top line and how does it fit into that NASH meeting schedule that we're seeing these days, I think that makes it special.
I agree with that, Jörn. I think that is one of the things that makes it special. The other thing that makes it special is that they make sure to incorporate discussions on regulatory. And last year, the regulatory discussions wound up really highlighting that both FDA and EMA were willing to consider the idea that you did not have to regress fibrosis to meet a fibrosis standard. And that was, I think, a newer thought. It came up a little bit at NASH-TAG, had not been around much in 2020, so that was a big deal. I'm going to be intrigued when we get to the end of this to see what they come up with this year. But thank you for the lead-in, and with that, let's dive in, because we have lots to cover, not a ton of time to do it. Who would like to jump first, preclinical or pathogenesis?
I'll dive in. So I'm really excited. I don't know if there's a specific talk, but I'm excited to hear more about updates on gut microbiome and the association with disease. I think it's a completely underexplored aspect of the human genome. It's often been referred to as the second human genome. if not even the primary, because for every one cell of human cells that we have in the body, there's 10 cells of microbes. So I really think that there is a world of opportunities to understand signaling and understanding from both diagnostics perspective as well as therapeutic. And we're really just getting to a time where we can do deep sequencing to understand the gut microbiome and the role in NAFLD and fibrosis.
Interesting. So there's one on nutrient-sensing IGF-1 signaling in NAFLD, the second day from Professor Chen at Berkeley, which looks to me like that would be on a related topic, maybe a couple of others as I flip along. A lot more actually on weight loss, interestingly enough. A whole bunch of stuff on weight loss in this meeting, but okay. Other thoughts, comments?
I mean, to follow up on Rachel's comment, I think that the challenge with the microbiome is always to understand its complexity and get a standardized readout within the clinical trials. And there's going to be some clinical trial data that's being discussed also. Becky Taub's going to discuss their phase 3 program. This is really the microbiome's exploratory scientific part, I think, of this. And we're not in the space to fully include that at more advanced stage clinical trials. However, it is extremely interesting. And just to follow up, maybe Rachel, your take-home disease activity biomarker or monitoring tool from your perspective, being deeply involved in genome analysis and diagnostics, where would this fit in just from a technical perspective? I mean, what can we achieve? What type of technologies do you have? What do you think would come in handy here with a microbiome?
Absolutely. So I think the first question we need to ask is, what is the standard microbiome? This is why I'm alluding to the idea that this is still very much preclinical and basic science today, because we don't even know what the standard is. And so when we can get an understanding of what the standard is compared to perhaps monitoring long-term outcomes, because your microbiome is dynamic, it's ever-changing, and I think that it might not be sensitive enough to be used as a staging tool or a diagnostic tool in the sense that a diagnostic tool in the sense of staging, but rather predicting long-term outcomes. So what are the pathways that are getting dysregulated as patients are healthy, patients with dyslipidemia, patients with early stage NAFLD. So I think that if I had to hypothesize, this could be a tool for long-term outcomes, but it's so early right now that there's nothing grounded to base that on. But that's where I see this going in that direction.
Sure. But I think then you really got to be talking about the cirrhotic population, which is more closer to outcomes. And there's some good data from the cirrhosis in general, not just NASH cirrhosis being generated, that they predict decompensating events, and you can modulate that with some of the therapeutics we're using in clinics today. I think here you're right.
Completely naive question. But if a lot of the places that have certain kinds of diet challenges around food insufficiency and where calories come from are in same subpopulations that tend to be defined as having PNPLA3 deficiencies or challenges, does that tie into this set of issues or does that go somewhere else?
I think it could, because in the sense when there's genomic rearrangements, that can drive disease. So for most of the last 50 years, we've only looked at protein coding genes on the human genome, and are really starting to look at these non-coding regions, which is 98.5% of the human genome. And when you have these certain high-risk alleles, they may or may not rearrange those non-coding regions, which has an influence on the microbiome. So those are absolutely associated. And I think we're just starting to ask questions in this capacity. So there's really a wealth of knowledge to understand. And I think that we're getting there with the cost of sequencing has come down so exponentially in the last 15 years that we really can ask these questions and monitor patients long-term to see what changes over time and what is standard, what is the norm, and what changes and drives disease.
Okay. Other questions or thoughts on microbiome issues?
I remember at NASH Tag, Scott Harris did a very interesting session, and I asked a question which wasn't raised in the room, but Scott actually contacted me, and I had a personal one-to-one session with Scott Harris discussing the effect of micronutrient problems in the rapid weight loss that they see. So I'm I'm looking forward to his session on day 2 at 3 o'clock, following up on the weight loss, because there is a number of micronutrient imbalances that occur in the rapid weight loss and how that they affect or don't affect patients in clinical trials. So obviously the gut microbiome is altered in that, and quality of diet we are seeing, but there was a bit of information out at EASL last year and ARSL that the quality of diet actually improved the liver steatosis in preference to weight loss. All of these, as has been discussed already today, shed new light on it's not always quantity of weight loss, it's actually the quality and how you lose it and what you lose it with, which may well benefit a lot of people. Now we can measure internal liver fat with simple tools like FibroScan, but MRI, PDFF, and all of the other measurements. If they don't feel that they've really got to bend over backwards to lose something that's unachievable, everybody's own weight loss, because of better technologies, better non-invasive, which we're going to discuss in this meeting as well, may well prove the key to success in each individual person's care pathway for losing weight and altering their liver fat.
Let me follow up on that. It's something that's been intriguing to me is, of course, the type. It's weight loss is the one thing, but the type of eating behaviors. And there's been a recent paper in Hepatology Communications, I think it was JHEP Reports, where they reported on the time-restricted feeding concept. Not so much about, absolute weight loss, but also changing the pattern of macronutrient or micronutrient uptake. And I think this also has an effect. So the complexity here comes in again. When do you eat? What do you eat? What are your germs doing with that? And what is the genome that you have and how to respond to it? I mean, this sounds very complex. I think in the end, it's not. It's so much body weight can be measured. We can measure, as you said, liver relevant or tools, diagnostics like liver fat, and that way we can counsel our patients. But the answer, in my experience, and maybe Ian can say something, it's very different from what a patient can do for himself. They start at a different level. Some have this type of nutritional problem, and they might need a different nutritional solution compared to someone who overfeeds on, let's say, orange juice, just to make an extreme.
It's a really valid point. The best mechanism for weight loss is what works for that patient. There was a paper just out in the New England Journal last week, a small randomized trial of time-restricted eating alongside calorie restriction versus calorie restriction alone, and there's no difference in weight loss between the groups. So it probably doesn't matter, to the whole population how you lose your weight, but to the individual it's really important. And some of that sort of personalizing message is, I guess, lost in those sort of aggregate studies, but working out what it is for the individual, having lots of tools in your toolkit to offer different patients different interventions is probably the key in the sort of personalized medicine approach to weight loss in the clinic.
Speaking as somebody who's lost a bunch of weight in his lifetime and then had to figure out how to keep it off and has finally done so, comedian Jackie Gleason once famously said he was the best weight loss champion of all time. He'd lost 1,000 pounds. It was saying 50 pounds 20 times. I've watched friends lose a lot of weight and gain it as fast as they lost it. So I increasingly think it's less about losing weight per se than figuring out what behaviors you need to overcome the systems in your body that are creating the challenge in the first place. And clearly that's going to be linked to all the issues, the physiologic issues we're talking about. But from personal experience, you don't know exactly what your physiology is when you're trying to lose weight in a way that you can keep it off. You might figure it out over time. I know some things about myself, but therefore it becomes, I think one of the reasons it becomes so challenging for physicians, as we talk about, is that, and back at Stephen's Peek and Shriek or Greet 'em and Street 'em, you don't have enough time with patients to learn the kinds of things that you'd have to learn or help them learn about themselves to be able to make that kind of process stick. I don't know that that's the province of a conference like this one. I think it's the province of a bunch of conferences and papers. But I think that's where a lot of that challenge lies. Other thoughts on this, or any other papers or thoughts from the pathogenesis and preclinical topics that are worth spending any time on?
I think let me just touch on the preclinical. There's a presentation by a scientist with Gilead, Aisha Oxley-Armlovich, and I said that wrong, and apologies for that. But they're discussing two-dimensional cell cultures and animal models to actually look at ectopic fat and fibrosis. And I think it's very interesting to see Gilead Sciences here discussing human precision cut liver slices and three-dimensional co-culture to study certain drug combinations and improve the understanding of cellular interactions. I think that type of modeling, and Scott Friedman, when he joined us here on the podcast, mentioned that once or twice, is particularly interesting because you can look at in the context of the liver under the microscope. You actually take a slice from a liver cultured ex vivo and then expose it to drug and see what happens. I think that's very hands-on, and it teaches us a lot about things that happen in the liver. Not necessarily the whole body, insulin resistance, what happens to the adipose tissue, of course, but what happens at the level of liver cells. And I think that's a very nice preclinical preclinical tool, and I thought that was interesting to have it in the program here.
Just following up from Jörn's and Jeff's conference in Barcelona, there's obviously Helena Cortez Pinto doing the NAFL pathway and the referral care pathway. So it'd be interesting to see if there's any difference within the sort of things that she brings up that have come out of the conference in Barcelona next weekend.
Thanks, Louise. I want to jump back quickly to Jörn's comment. Jörn, when I read that brief description of the talk, what I was interested in as much as anything else was the last sentence, and I wonder how deep she's going to go into it, because she says not only is she going to share the case, right, the Firkestad effects are case, but then she said to comment on the potential for translational models in ex vivo translational models. I'm wondering what she'll be comfortable saying about that and what we can learn from that statement, because that's another area that isn't particularly plumbed yet, I don't think, at least what I read. A lot further you can go with it.
I think that's the critical point, though, isn't it, Roger? The animal models can only take you so far. Jörn's published about why trials fail, and one of the reasons is that humans are different to mice, and a human precision-cut liver slice is closer to a whole human than perhaps a mouse is, but it still lacks a lot of the other critical aspects of pathobiology, as Jörn says, everything outside the liver, functional immune system, and everything else. In some ways, you might prefer a drug combination that had a clearer signal in a clinical trial to be able to really understand what a true positive drug impact was in a precision-cut liver slice rather than the combination that they're testing. But I guess it'll go some way to try to understand how directly translatable outcomes are in that model to future clinical outcomes and trials. You're frowning. It looks like I've confused you.
No, I'm thinking. But what I'm thinking is, I'm going back to some of the conversations we've had. There's a real challenge with combination agents, right? With figuring out how you get from the hypothetical to the real. In some ways, that might be more of a challenge than it would be with a single agent. Single agents are just cleaner to figure out. So I take your point that might not be the perfect way to do this, but I think it was Churchill who said about democracy once, it's the worst system we have except for all the others, or something equivalent to that. And I'm wondering whether ex vivo for combination therapy would be a similar thing, which is that it's a way of getting at some of the complexities that you'd have a hard time sorting out otherwise until you ran large trials. Again, I'm not a trial person. If any of you can just tell me that was silly or gracefully correct me, but that's just how it struck me when I looked at it.
But I mean, I guess there are some human trial data for those agents in monotherapy and in combination, and experimentally I'd be very surprised if they don't present data for each of those drugs alone and then in combination to try and tease out the effects that you can see ex vivo and then the effects that you observed within the trial framework. I mean, it's a small trial, not necessarily easy to extrapolate the results, but still one would hope that you might be able to learn something from what they've done in terms of the translatability of that model.
Yeah, I agree. And some of those are in clinical trial, and the data has been presented by Naeem Al-Khoury previously, combination with GLP-1s, for example, the drugs they're exploring or they have in their abstract there. I agree there's more to be learned from the clinical trials, but here at the mechanistically level in the liver cells, I think that's just an interesting abstract and topic.
Actually, Naeem and Mazen published on that, or they had publication actually published last week on one of the aspects of the triple study that I'm thinking we might decide to do an episode on at some point in the spring. I can point to one more I'm kind of interested in, which is the SomaLogic presentation, the first morning on liquid liver biopsy. The reason I'm intrigued is, if I recall correctly, SomaLogic did better, and was it one of the litmus, where SomaLogic clearly turned out to be the most robust of the systems they looked at. I'm not sure what this paper has to do with any of that, because it's not crystal clear to me exactly what he's going to talk about, but I don't know nearly enough about them. So I'm looking forward to having the opportunity to learn from that paper.
Yeah, the fascinating thing about the proteomics analysis tools is that it gives us such a wealth of biomarker and then comes up with a final score. And that has been, interestingly enough, pretty robustly done so in litmus. And we're going to see more data from the clinical trials because it's run along this. And I mean, maybe Ian has also a thought on this, because if we think about what we're doing today using FIB4 to rule in, to rule out, this is such a biomarker that captures so many different aspects of the disease. And sometimes the challenge is in how you get a surrogate score and how does that all come down in your patient, how you're going to use that data. I think as a treatment response marker, that could be one way forward. As a cross-sectional diagnostic tool, we're going to see more data on that, bottom line. But again, the complexity of this biomarker is just very high with all those proteins that are being explored in that multi-omic approach.
I think that's right. One of the things that we're missing is the NASH response biomarker rather than we're missing lots of response biomarkers, but a NASH response biomarker, indeed diagnostic, would be really helpful. And it may be that that's where it might find its place there. One of the issues is the complexity, and trying to distill it down to a perhaps more palatable number of molecules that can be more readily understood might help in terms of at least conceptualization and people's understanding of what it's doing. But I think it's definitely an interesting program that they've got, and the data, as you say, are in litmus are very strong.
But then on the other hand, scientists like Rachel can teach us. There are many more things than a physician's simple mind can understand. We look at one biomarker and try to link that to the patient. Of course, the complexity of the technologies and the biomarkers that are being in clinical development is much higher. And maybe we do not we have to clearly link it to the disease. I think that's important, and we're learning more. And maybe this talk is going to educate us on this more. And in an ideal world, we'll link it to an outcome. Okay, so we're going to not be interested in liver biopsy at all, but we have this biomarker, and then see what happens to the patient. Prediction, as Rachel said, maybe this could be held up for microbiome in cirrhotic patients, or it could be such a biomarker as he presented here, but then the somologic analysis, and then we can make calls on what would be the right path forward for that patient.
Yeah, and let me follow up on that just from a general standpoint. I think that it is of vital importance that we take an integrative genomic approach. So we don't just look at the transcriptome and derive insights from just looking at RNA sequencing data, but rather looking at the transcriptome and various aspects of the human genome, such as the epigenome and more specifically the methylome, and ensuring that these aspects occur concurrently, because we all know that NASH has a high degree of heterogeneity. So a gene upregulated in one patient could be downregulated in another. So when you're taking a multiple omic approach, you can look at what's truly occurring concurrently and then derive insights for clinical outcomes, for diagnosing, prognostics, and so on. So I think for a long time, we've approached these tools quite simplistically. So it's nice to hear that we're being more strategic in understanding their outcomes. With that, Roger, if I can switch gears, and I wanted to mention another talk that I am interested in hearing. On day 2, Stefano Gianni Corradini from Rome, Italy, is giving a talk on liquid droplets and epigenetic susceptibility in NAFLD. And they're looking at different types of static hepatocytes based off of size of droplets and epigenetic markers associated. So I'm really excited for this talk. And I really think that the epigenome is starting to emerge front and center on susceptibility of disease, as well as predicting long-term outcomes. And I think it's about time that we use these tools appropriately. So I'm excited to hear about this talk. And what's interesting about this is it looks like the studies that he has published are directly from liver tissue data. And I'm excited to see what that looks like, because then we can start to quantify what's going on directly on the liver. And then next steps, which I'm personally excited for, is can we quantify these markers in circulation, and how? So he talks about different microvesicles and macrovesicles and the size quantification of each. So I'm interested to see what he proposes for next steps from these findings.
So Rachel, that's great. We're going to lose you in a minute or two. I know. I want to ask you one question real quickly, and then let everyone else comment on that. This is a slightly different question. I want to raise it. Going back to Jörn's comment, how much of the science at the level you're talking about do you feel physicians need to understand? A, physicians need to understand in order to be able to treat, and B, physician researchers need to understand in order to be able to do a better job on drug development and assessing what the results of their studies mean?
Yeah, I mean, I see this often. I work with graduate students in the DC area, and there's so much happening so fast that I think that physicians need a crash course every 6 months to understand what the new and most effective methods are. And without those crash courses, or at least spending a weekend really understanding what has changed in the last few months, we're limiting what we can do for patients. Because if we're just using epigenetics as a genetic susceptibility, we're missing out on a world of opportunity, everything from diagnostics to therapeutics. And I think it's just, there's such a limited and lack of communication between the clinical world and what's really going on the grounds in the research world. So I think several crash courses are important multiple times a year, because things are changing rapidly. Even as a geneticist myself, I get lost in the complexities. And I think that if physicians could just at least actively reach out I don't know what that looks like. I don't know if there needs to be specific courses, but I think we're missing aspects of the conversation right now. So we need to do better. I don't know what that looks like, but that's a great question.
I'm not sure how often I would need that, but sign me up, Rachel.
Yeah, that was kind of what I heard, Jörn, between you and Ian on how much of it do you need to know? And the answer is some of it, not all of it. You can't keep up with it fast enough. But other than that, Rachel, thanks. I know you got to step out for a bit and you'll come back on closing question, but thanks for joining us. So, Ian, I don't know how satisfying you found that as an answer to the general question about how much of this people need to understand. I'm intrigued by your thoughts.
Well, you could spend 5 years at medical school or longer in some systems or less, learning a lot about pathophysiology, pharmacology of lots and lots of different things. But you forget quite a lot of that. And as you practice, particularly if you practice widely in general internal medicine or in general practice, then I guess that you forget quite a lot of that and you understand what works, what test to order when. As a general practitioner, I might not need to know where BNP comes from, but I know that it's a diagnostic test for heart failure. And the question is, which communities need to know what? I think it's a real challenge with emerging technologies for people to learn enough so that they're knowledgeable about them so that they can actually understand where they might be useful in their practice. That's quite a big ask for the sort of general secondary, even tertiary care, clinical gastroenterologist, hepatologist. That would be a big thing to learn, a big step to take, particularly if you're being asked to take a similar step in new pharmacological agents, in new approaches to endoscopy, which might all be part of your clinical practice. All of these emerging fields have to be developed by clinical and academic and industry leaders, but with a clear eye on where that's heading. And I think, my comment about distilling down and making it simple, having composite biomarkers of lots and lots of different things that add incrementally on top of established biomarkers, whether that's FIB4 or something else in the future. The question comes then, what will the value of that be in primary care or in secondary care practice, and at what cost? Because ultimately it's those things that will decide how those tools are taken up before they become very, very widespread and inevitably very, very cheap.
Yeah, and just to add my thoughts to that, Roger, we are at the point because because of the interaction of clinical translational scientists and basic scientists in the field, and during the development of novel drugs, diagnostic tools, there's a big need to exchange. And there's been from my perspective, it's been a tremendous wealth of learning, being able to talk to basic scientists or people like you had on the podcast here, Scott, to get to know his ideas and then link it back to the patients we're seeing in clinic. I think during that development progress, it's very important to get the depth of all that information. In clinical practice, in the end, I think as Ian said, if they know there's the test that's linked to a certain outcome, that becomes less important. It has to be linked to the disease pathophysiology for the physician to relate to it as a disease driver, diagnostic marker, or something like this. But in the field of clinical translational science, like Ian and I am involved now, we've learned so much from the interaction with the basic scientists, and that helped us to come up with hypotheses and formulate ideas.
I was only going to comment on the SomaLogic one, in the fact that interested me more from the continual link to diabetes and heart disease, in the fact that the only way we're going to drive awareness of liver-related care is to drive it within the two big or considered big fields of cardiology and endocrinology, despite liver disease being outnumbering those patients currently. So that's where the strength lies. The more that we can tie all of this into those diseases and the more we can offer to prove the strong links between the cardiovascular disease. So I'm interested to see his figures and what comes out and what the actual strength of that is in the cardiovascular risk. Because I did this conference a couple of years back. It was the last in-person conference I did. And in fact, there was a session on fatty liver disease as a cardiac disease. So that area of driving it out, I think, is a strength.
Yeah, I remember your comment at the time or last year that the strength of this Congress was that, in fact, they took a look at things in a more interdisciplinary kind of way. So let's shift gears into the other areas, drugs and development, diagnostics, and what we're learning about patient treatment in general. There's a wealth here on weight and a bunch here on drugs. So I'd be interested to hear what any of you found particularly interesting or compelling on those topics.
I just went through the program to make sure I'm not missing anything. It's pretty much that's what I meant in the beginning. It's chief medical officers or key people from the companies that are reporting on their drug development program. There is Galmet present with aramcol. There is some data of semaglutide for combinations. There is the cirrhosis study that's been done by Gleason, and Paul Boots is going to talk about belapectin as an inhibitor. So the abstracts don't really give away the topics they're covering, but it's very interesting to hear from them what they think has been a breakthrough for their drug development program, what's been helpful, what maybe they will mention things that they had to overcome to be successful in enrolling patients and ongoing clinical trials. I mean, they can't say much about the results, but it's very interesting just to hear from their perspective what's important for the field and how we can move this forward. Because at this point in time, we've seen some failures. These companies really come up with strong candidates to be explored in clinical trials, and one of them might make it over the finish line, which we hope and I'm confident of. And just to hear their perspective on this path is very interesting.
Yeah, I agree with that. In the making it over the finish line, it'll be interesting to see if Becky Taub shares anything on phase 3 resmetirom that isn't widely known yet, or is able to talk about it in a way that isn't widely known yet, in terms of who might get over the finish line first.
And something that the Madrigal team actually has been very good about is actually to educate us on the NITs at the screening and baseline populations. And in addition, they have some open label data that they have discussed in the past. And as such, the drug development program is pretty big and has helped the field to understand and correlate to certain NITs. That's interesting there. I think she might have some of that in her talk. Well, my hope would be she does.
So, Louise, any thoughts on Scott Harris, given that you mentioned your private Q&A with him before, or on his talk?
No, I'm just looking forward to what he's got to say, because I think he follows on from Anya Geretz on day one, which is looking at the bariatric surgery and looking at the rapid weight loss. So it'll be interesting to see what he has to say for that, but it does interest me. Just jumping on what Jörn was saying, there's, in fact, Anna Geretz is on the second day, but it's the semaglutide data. I'm interested in that because, of course, that's a drug that's in the real world. It's been approved by the UK for use by NICE. So this is an area where we could be getting really real-world data in a lot of countries with use of non-invasive technologies in patients with multiple other health conditions, and I don't think we're collecting that data. I'd like to see, obviously, liver non-invasives as standard of care in these patients. So it will be really interesting to see what the data from this trial suggests, because it's looking promising and it's already in the real world in obesity and other areas. So let's see.
So it's interesting to me when you pair that paper with what Scott Harris is going to talk about. I think Altimmune has been comfortable talking about comparisons with other agents in clinical work to date as they've gone along. I remember at NASH Tag a couple of years ago, certainly, a paper that talked about that a bunch. So they'll be looking at monotherapies and some dual agonists, all of which are primarily, either GLP-1 or GLP-1 GLP-GIPs. So it'll be interesting to see how that stacks up or contrasts to what Novo Nordisk will be talking about, which is semaglutide and a different kind of combination therapy, right? FGF21 or something that works in a very different way. I'll be intrigued to see those two talks and how they compare and what we might learn from one or the other.
Yeah, I think comparing and contrasting in that way the nutritional aspect and weight loss, and there's a couple of sessions on weight loss with the drugs, it's almost a head-to-head comparison in the room.
There are two more drug development programs that are being presented. You mentioned Madrigal before, but one that we should touch briefly is, of course, the Hapion, CRV431. They've had a positive phase 2A program, and that informed a phase 2B study that's ongoing now in patients with advanced fibrosis. So the concept of cyclophilin inhibition in NASH is interesting, and we'll see what their take is on this. And of course, the second one, and I thought that was interesting too, is the French company Poxel that developed pioglitazone, that is an isomer or in a certain way modified to try to avoid some of the side effects we've been seeing with pioglitazone, namely weight gain. And interesting how that comes in, because pioglitazone, we know, has been showing effect, particularly in patients with diabetes. The famous PIVENS trial wasn't positive, but there's a lot of subgroup analysis been done subsequently. So the PPARs as a class, of course, have been around, and it'll be interesting to learn how they plan to modify that and to get some of the unwanted effects out of that drug class.
No, I mean, I think the session I think is interesting, as Louise has alluded to, is there's clinical data about weight loss being presented from Oxford, where they've systematically reviewed the impact of low-calorie diets on NASH and the various endpoints. And that paper's recently been published in Clinical Gastroenterology and Hepatology. And they've also got some preliminary data about a pilot trial of the safety and potential efficacy of a very low-energy dietary intervention, like there is for diabetes, and a program now in the UK that's being rolled out by NHS England. What's interesting is if you go back and look in the original reports of the very low-calorie diet, you see marked decreases in the small number of patients who had MR spectroscopy at baseline looking at liver fat. The dietary manipulation for weight loss will be interesting, but the parallels with the pharmacological interventions for weight loss that will follow from Scott Harris, I think, will be, it makes the case for pharmacological intervention, he says, having not seen the talks, but being familiar with some of the data. That's probably the standout hour for me, would be looking at that. It's just unfortunate that it's at the same time as the discussion from Valerie Paradis about HCC and NAFLD, which I think is an important side conversation because it is a clinical event that occurs in a number of patients that seems relatively unpredictable, even amongst the patients with advanced fibrosis.
I think the interesting thing is also the sessions on weight loss, with or without, and particularly without the pharmacological intervention, can address some of the questions that we have about the placebo lines and how people lose weight, what they do, how they change. And I think always understanding the control or the placebo group and their behaviour, as we've alluded to many times on the podcast, it has the ability to derail a trial very much or prevent a drug going to getting approval. The more we can understand how diet and lifestyle affects in the world of drug and pharmacology, the more we might understand how our placebo arms and how we can manage that in future trials.
It's an interesting point, that, Louise. One of the issues with the approach that the Oxford team have taken is it's trial level data. So you look at trial level changes in body weight and try and correlate those with outcomes. And that's probably insufficiently sophisticated to try and do the type of analysis that you're suggesting, where individual level patient data would be much stronger. What's apparent when you look at the manuscripts, and I'm sure when they present the data, you'll see that the net weight loss to predict a 20% improvement in whatever outcome, whether that's NASH resolution, 2-point improvement in NAS or fibrosis improvement, the net weight loss to achieve that is 0, because that is the placebo response. People have heard my views previously, but that placebo response relates to the limitations of histology, specifically sampling variability, and additionally probably a bit of observer variability. But I think it's predominantly sampling variability, and the sooner that we accept that and stop trying to fix it using tools that are not going to fix it, the better. It may be still unproven. I think that AI, and the talks about AI here, that AI will impact on that, because you'll get a better, possibly more holistic assessment of fibrosis or of NASH, but I'm not convinced. And if you take the same size sample, 20 millimeters in length, 15 portal tracks, and use that as your baseline and end of trial assessment, then I think you'll struggle to get away from that 20% type rate. It's consistent across all of the trials, and as I say, very likely to be explained by sampling variability rather than anything else.
Nothing much to add beyond maybe highlighting that, yes, there are 2 talks in that conference trying to bridge that gap, at least from the perspective of human variability in assessing those changes, using AI to more robustly come to an objective readout of that piece of liver tissue we're taking out. And again, I think the field has made a strong argument to move away from liver histology as a surrogate and look at outcomes, look at NITs to support those outcomes. And we're on a good way, I would say. And based on what Ian said, and he highlighted the limitations nicely, this is so important. And the 2 AI talks here, one I think addresses imaging, and the other one, which I'm not too familiar with, the liver histology, will be interesting to see. I think we're most advanced on, of course, the quantification of objectively quantifying fibrosis rather than other changes that are more diverse or inconsistent, but that will help us tremendously.
That all makes good sense to me. I mean, the degree to which it's an issue of sampling variability is the one thing you really can't fix, right? Without leaving histology behind. AI can fix inter-reader reliability, but if it's purely an issue of sampling, you can't fix that. I suspect one of the things we'll learn as we go forward is exactly how much is fixable without getting into sampling variability. But Jörn, I think big question, as we talk about frequently on the podcast, is going to be how quickly will we be able to step past histology entirely, right? And some of that will be about how good the work of the consortium, particularly I think Neil NIT, which is dedicated to that specific issue, how good all that work will be from all the consortia. And the second is, was the work gets done, what does it prove?
Yeah, agree.
I think that's the multi-billion-dollar question, really, in terms of, because I think it'll have a lot to do with how quickly can drugs get approved and how quickly can diagnosis spread out beyond specialty specialties, into primary care, or into environments that have less money to spend on diagnosis and less time to do it. So I think you're really kind of at the heart of the issue there.
Yeah, for the timeline, I think the good news is the more advanced patients that hit those endpoints, we're going to have NITs that we collected already in these consortia. We're going to learn which one's going to develop outcomes. And there have been some publications, for example, the cirrhotic simtuzumab or the Stella 3-4 from the Gilead trials that have educated us on changes in NITs, simple NITs, but they're linked to events in admittedly very selected patient population. So I think in more advanced cases, we're close to that. I would say 3 years timeline, and it might take a little bit longer. But if in the meantime we get a positive surrogate endpoint in one of the phase 3 clinical trials, that would help, of course, tremendously to then also move this into real life and understand those changes outside of clinical trials.
I was going to say, Roger, I mean, this whole discussion takes us back to the thing that we missed out at the top of the agenda, which is the EMA's perspective. And in contrast, I think, to the FDA, the EMA, at least to this external observer, seem rather quiet. And their last published position paper is several years old now, and we don't see them as frequently at meetings as we see the FDA, and that might be to do with the way that the meetings are coordinated. And I think it'd be very interesting to hear what they've got to say, at least in this forum, about how their view might be changing in the light of published data such that Jörn's just referred to.
Thank you, Ian, because that was the place I wanted us to wind up, was on that particular set of issues. I'm struck, I mean, last year somebody from the FDA was here as well, and this year we don't have that. What we have an EMA talk, and we have someone from AstraZeneca talking about regulatory pathways of NASH, and there's no comment on that talk at all, so we had no sense really whether Mr. Torstensen is going to be talking about FDA, EMA, whoever, right? We just don't know, number one. But number two, last year's talk I thought was interesting for the convergence, because it was the last time we saw the two agencies together. Well, I take that back. We've seen the agencies together since then, but at that talk they seemed to be highly convergent. They really were on a lot of the same topics, and as you point out correctly, FDA stepped out in public and moved past that a good way since then. So it'll be interesting to see what EMA has to say.
It'll be interesting to see who sits on the roundtable discussion for the liver biopsy as gold standard, because there are several pull-out sessions later on that day from the EMA comments. So it'll be interesting to see all of these rooms, all of these tables, correct me if I'm wrong, Jörn, are all in the same room. So that might get a heated discussion in one table. I'm not too sure which table I'm going to sit at yet, but there's several that interest me. I might earwig into several of them.
I would love to see if they could put tables 2 and 4 next to each other, the liver biopsy, the gold standard, and use of non-invasive biomarkers. And then on the other side of the room, they should put maybe 3 and 5, liver and NAFLD and insulin resistance and diet and lifestyle on NAFLD NASH. And clinical trial design could be in a third orbit all its own. And then what we'll do, if I'm there, and I think that's still an open question, I know Louise will be there, you can pick one, I can pick the other one. We will put one earpiece going in each direction and see if we can keep 2 streams of conversation in our mind at one time. That'd be kind of fun.
Now that's technology for you.
Or making it up as we go along and bluffing later. So in closing, I think what I'd like each of us to comment on is the one thing we believe we might take away from this meeting and the one thing we hope we will take away from this meeting. If you will, the practical case and then the optimist case. Brave one, go first.
Let me start. I think, as I mentioned in the summary of the talks, I'm very excited to hear the perspective of the different drug development programs and their medical lead or people that represent them, and hear about their thoughts, the opportunities, the challenges during running their clinical trials, and how maybe their phase 2A informed their phase 2B moving forward, because there's still been a lot of trials and a lot of attempts to develop drugs, and there's been positive phase 2, then disappointed in phase 3. So there's this critical momentum. How can you increase your chance of success when going into a large venture like a phase 3 trial? And I think just to hear their thoughts and how they went about this is something that is very interesting to me.
Thanks. You're next.
I'll go next. I think for me, I'm particularly interested in obviously the lifestyle management and the dietary ones, because those are the ones we can immediately put into care pathways. The drugs in development are further down the line, and how we adapt nursing care and delivery of services to patients will be designed as they roll out. There's obviously the conference next week, and then there's some sessions in here about how that may well adapt. That's what I'm looking for here, and listening to Scott's work, looking to the Oxford paper that's coming out. But also what I hope to definitely take away is things that I can use now about dietary modification, types of diets, and some of the data that will come out that can be implemented now in patients with more severe disease to sort of that stabilisation that's beneficial and that we've got actual data on now.
Okay, Ian?
Like Louise, I'm quite looking forward to learning more about the diet, but the perspective from the EMA, I think, will be very interesting to see how far they think that they can move. And I guess if you compare the original positions of both agencies, the EMA were perhaps a bit more conservative than the FDA were, and that will be interesting to see whether they're prepared to publicly state movement at the same rate as the FDA have done to date. And I think that would be an interesting discussion.
One thing I'm hoping to learn and hoping to see is that in the biomarker and non-invasive diagnostic track, that there is an integrative diagnostic approach. So in the sense that if you're looking at serum or image-based biomarkers, that you're not just comparing it to the imperfect standard of liver biopsy. So for the development of new tools and new biomarkers, we're looking at liver biopsy, perhaps in the presence of artificial intelligence, and then comparing that to various imaging modalities, whether that's MRE or MRI, transient elastography, FibroScan, and in the presence of serum markers to develop new tools to make sure that there's an integrative approach. So I think that if we're moving away from liver biopsy, we need to be intentional in our strategy and our methods to ensure that the patient samples we're utilizing are well characterized. So I really hope to hear about that in the methodologies of those tracks.
Agreed. So I'm going to pick kind of a wild card that we've not talked about very much today. But last summer, we talked about the idea that drug development was more likely to come faster in cirrhosis than it was in advanced fibrosis, A, because of the need, and B, because you didn't have to worry about endpoints quite as much. Now, some of that's changed as FDA moves more towards NITs. But there are several talks in here that come at cirrhosis from different, starting with Sven Frank's talk at the beginning, where he specifically says, I want to comment on what clinical trial design should be for cirrhosis patients. And what about the vascular alterations lead me to think that. And then I guess, Jörn, you mentioned the Galectin paper from Paul Budas, and there are a couple of others in here. I'll be interested to see what we learn about cirrhosis as a development path, how easy it is to figure out what to do, and how well along that path people are to get there, because I think that will be an interesting commercial issue.
Yeah, and just to follow up and link that, I don't think we're exchanging outcomes with NITs. It's that we're learning how to link the NIT to the outcome and dissect that from the histology maybe. And really, I'm going to say this because Donna Cryer used it, to move beyond the biopsy is the term here.
Well, I guess what I meant when I said outcomes is that histology, you need subpart H outcomes before you can actually get the clinical outcomes, whereas with cirrhosis, you go straight to clinical outcomes because subpart H doesn't matter because regressing fibrosis doesn't help. Sorry I didn't state that better. With that, let me say thank you to Ian for joining us, and Jörn and Louise. We'll be back next week with a fascinating, very different kind of session, which is we're bringing in Andrew Scott from Global Liver and Achim Kautz from Germany, and maybe one more, and we'll be talking about all the challenges of patient advocacy today, the interplay of regulatory and legislative and community and corporate and everything, and how those pieces come together to make patient advocacy a more effective tool. It was an idea that Jörn suggested when he came on board, and I think it's a fantastic episode, and we're really fortunate to have these two people, maybe one more, to come talk about it. So with that, let me wish everybody a great end of April, because by the time we get back together again, it will be May, actually, won't it? This year's flying by, folks. So thank you very much, and I'll be back with the business report in a couple of minutes.
And now for the Season 3, Episode 22 Business Report.
It pains me to do this every week, but I want to start by saluting Ukraine and the people who are fighting so bravely there. Slava Ukraini, Heroyam Slava. Glory to Ukraine, glory to the heroes. One day maybe we won't have to talk about that anymore for a good reason. There's not very much business news today. Let me make that as quick as is reasonable to do.
The rest of the second quarter schedule is taking shape.
Next week will be something we've never done before, a session focusing entirely on the mechanics of patient advocacy from the patient advocate point of view. Andrew Scott will join us from Global Institute, along with German patient advocate Achim Kautz, and possibly one more. Not sure yet. May 11th will be our follow-up of the Innovation in NAFLD Care meeting in Barcelona the previous week. May 25th will be a session with Quentin Anstey and Stephen Harrison, discussing one or more NIT issues emerging from the Litmus Consortium meeting earlier in May. The week of May 18th and the week that has June 1st will include 2 regular episodes episodes and a special. One episode covering 5th Global NASH Congress, another previewing the Global Liver Institute's International NASH Day, and a third containing the interviews we've promised with Lars Johansson and Mike Patel, along with Stephen Harrison talking about drug development and possibly a mystery guest to be named. The first full week in June focuses on International NASH Day. The second full week will be our preview of EASL at the end of the month, and the third full week actually heads into the EASL coverage. Somewhere in there, we believe there will be a major news story. We will find a way to fit it in.
The buzz keeps growing as we hit 1,000 downloads for the 8th week in a row and 5,000 downloads over the preceding 28 days for a second straight week.
This marks our 8th straight week with over 1,000 downloads, our second with 5,000 downloads over the previous 28-day period, all in what's supposed to be a down season for this kind of a podcast. Normally, we do best when there are major meetings. There haven't been any recently, and as the meetings start to pick up again, things should only get better.
And in the vault, something's old, something's new.
Season 3, Episodes 10 on screening and treatment pathways in the US, UK, and Germany, and Episode 14 about balloon hepatocytes have stayed strong, actually grown stronger in the past week, as does the renowned Season 2 Conversation 46.2 with Stephen and Jörn summarizing basic science issues from Paris NASH. New items in the vault include Season 3 Conversation 11.1 with Stephen Harrison and Mazen Nuruddin describing the thinking and motivation that led them to organize NAIL NIT, and Conversation 17.1 with Scott Friedman describing the path to the CAR-T/mRNA therapy for antifibrotics. Now, Scott has written an eloquent summary of this work for the New England Journal of Medicine recently, which we plan to post on the Surface NASH website as soon as we can. From Season 2, 2 other conversations did well. 54.1, which features Donna Cryer and other patient advocates discussing their reactions to last November's patient-focused drug development meeting with FDA, and 57.1, which reviewed NIT presentations and posters from last fall's AASLD meeting. Any week we've got 1,250 downloads. A lot of other things are getting at least a little bit of attention, so check it out for yourself. With that, I'm done for today. I want to thank our team, Magic Mike, Eric, Steve, Murph the Magnificent, for a typically tremendously productive week. Even more than that, I want to thank you, our listeners, for giving us the motivation to go on and your increasing interest over, as I point out, what might be a down kind of a period in other years. And with that, I'm off. Enjoy your week. Enjoy episode 22 and the conversations. We'll be back next Monday to talk about patient advocacy. And then on Wednesday, I'll be flying off to Barcelona for the Innovations in Natural Care Congress. Very exciting. So for you, for us, everybody, if you're in the Northern Hemisphere, enjoy the spring weather. Stay safe. Surf on. Look forward to seeing you all next week on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com and we will answer on the podcast or the website.